Okay, so welcome to this next video in which we are discussing type 2 activation of endothelial cells. So we've discussed that when there is a pathogen that is infecting a portion of your tissue, such as this Staphylococcus aureus in your skin, then what's going to happen is that pathogen-associated molecular patterns associated with that Staphylococcus aureus are going to be recognized by pattern recognition receptors on the sentinel cells, which are these resident macrophages and also these dendritic cells. And what's going to happen is that those resident macrophages and those dendritic cells are going to become activated, and they're going to start producing pro-inflammatory cytokines. And the two main pro-inflammatory cytokines that they produce are interleukin-1, IL-1, and tumor necrosis factor alpha, TNF-alpha. These two pro-inflammatory cytokines will go to the endothelial cells of the arterioles, the capillaries, and the venules that are within your affected tissue, and they will cause type 2 activation. The interleukin-1 will bind to the type 1 interleukin-1 receptor on these endothelial cells' basolateral membrane, and the tumor necrosis factor alpha will bind to the tumor necrosis factor receptor 1. Both of them will then lead to the assembly of these signalosomes, okay? So in the case of tumor necrosis factor alpha, it binds to the tumor necrosis factor receptor 1, and this then activates this TRAD protein, which stands for tumor necrosis factor receptor associated via death domain protein, okay? The TRAD protein sticks to the intracellular aspect of the tumor necrosis factor receptor 1, and then another protein sticks onto the TRAD protein. Okay, that next protein is RIP1, which stands for serine threonine kinase receptor interacting protein 1. Okay, then what happens is another protein is recruited onto that serine threonine kinase receptor interacting protein 1, and this protein is by the name of TRAF2, standing for tumor necrosis factor receptor associated factor 2. Okay, so that's the assembling of the signalosome for tumor necrosis factor alpha. For interleukin-1, what happens is that two proteins associate to the cytoplasmic domain of the type 1 interleukin receptor. Okay, and one of these is the myeloid differentiation primary response gene 88, or MYD88. Another one is this Toll/interleukin-1 receptor accessory protein, okay, or the TIRAP protein, also known as the MAL protein. Then what happens is this myeloid differentiation primary response gene 88, MYD88, associates with two more proteins, namely the interleukin-1 receptor associated kinase 1, IRAK1, and the interleukin-1 receptor associated kinase 4, IRAK4. Then what happens is another protein binds onto the IRAK1 protein, and this final protein is TRAF6, okay, standing for tumor necrosis factor receptor associated factor 6. So here's this signalosome for the interleukin-1 receptor. Okay, so now what will happen is that both of these signalosomes are going to do the same thing within the endothelial cells. They are going to lead to a whole downstream cascade of kinase activation, specifically of the mitogen activated protein kinase family. Okay, but the overall outcome of these signaling pathways is that you get the activation of the activator protein 1, the AP1 transcription factor, and also you get inhibition of the inhibitor of kappa B protein, so that it will release the nuclear factor kappa B, which remember is this dimer of the P50 protein along with the P65 protein. So now the overall effect that all of this has come down to is that we have activated two transcription factors. So let's go over the page and talk about what effect these two transcription factors are going to have. So we have activated the activator protein 1, AP1, and we have also activated nuclear factor kappa B, which, remember, consists of this P50 protein bound to this P65 protein. Now these are both what are known as transcription factors, so let me just remind you of what a transcription factor is. So if we have a piece of DNA here, basically in all eukaryotic cells, upstream of every gene that is within the DNA of those eukaryotic cells, you have what's known as a promoter region. Okay, now this promoter region is not part of the DNA that will actually be translated into protein. However, it is very important for controlling the expression of the downstream gene. So let's say that this box that I've drawn here is the gene, okay, and I'll highlight that in a certain colour. So I'll highlight this in purple. So this is the gene here. And then upstream of the gene, you have a portion known as the promoter region. So let me put that here. And as I say, this promoter region is not involved in actually making the protein. It's not going to actually be translated into a sequence of amino acids. The gene is what's going to actually be translated into the protein. However, this promoter region is extremely important for controlling how much of the gene product of the gene you're actually going to make. And the reason is that if you think about it, in order to make the gene product of this gene, what needs to happen? The RNA polymerase enzyme needs to come and bind to the DNA, and it needs to start working its way along this gene and producing a piece of complementary mRNA that's complementary to the coding strand of the gene. Okay, this mRNA will then be translated by a ribosome into a protein. So really, how much protein you're actually going to produce from this gene is determined by how much mRNA you produce, okay? And how much mRNA you produce is dependent on how much the RNA polymerase is actually going to come and bind and transcribe this gene, okay? And basically the region where the RNA polymerase binds to is this promoter region that's upstream of the gene. So the RNA polymerase will bind to this promoter region, work its way down, and produce the piece of mRNA that's complementary to the coding strand of this gene. Okay, so that's how the promoter region controls the expression of the gene, because if the promoter region is really good at binding to the RNA polymerase, i.e. if it's got high affinity for the RNA polymerase binding, then the RNA polymerase will bind a lot, and that means that you'll get a lot of the mRNA for this gene being produced, and if you get a lot of the mRNA being produced, then you'll get a lot of protein produced. Whereas if the affinity is very low for RNA polymerase, then RNA polymerase will hardly ever bind, and that will mean that you won't produce that much mRNA at all, so you can't therefore produce that much protein. So that's how these promoter regions control the expressions of genes. Now, a transcription factor is something which will bind to the promoter region. So here's an external chemical species. They're usually proteins, but I don't think they have to be proteins. Okay, so a transcription factor is some molecule which will come and bind to the promoter region and alter the affinity of that promoter region for binding to the RNA polymerase. So let's highlight this transcription factor in bright green here. Okay, so this will bind to the promoter region and it will change the affinity of that promoter region for the binding of RNA polymerase. And you can have transcription factors that increase the affinity, and they're therefore going to increase the transcription of the gene. So they are enhancers. They're going to increase the protein expression. And you can also have transcription factors that are going to decrease the affinity of the promoter region for RNA polymerase, and they will be repressors. They're going to decrease the production of that protein associated with that gene. Now basically, the activator protein 1 and the nuclear factor kappa B, the NF-kappa B, both of these are transcription factors. They're going to bind to the promoter regions of certain genes and increase the expression of those genes. Okay. And the rest of this video, what we're going to do is we're going to talk about which genes the AP1 and NF-kappa B are going to increase the expression of, and how this is going to affect the cell. Okay, so one of the starting genes, the gene that we're going to start off with, that's going to have its expression increased, is a very, very famous protein. It's called COX-2, which stands for Cyclooxygenase 2. Okay, now what does Cyclooxygenase 2 do? Well, Cyclooxygenase 2 is involved in converting a fatty acid, okay, so a long-chain carboxylic acid known as arachidonic acid, into prostaglandin H2. Okay, so to understand this, we're actually going to have to talk about something that occurs in type 1 activation of endothelial cells, okay? So in type 1 activation of endothelial cells, okay, so when the histamine activates the endothelial cells, one of the things which happens is that you get activation of an enzyme known as cellular phospholipase A2. Okay, so let me discuss this. Cellular phospholipase A2. Okay, and this is often abbreviated to CPLA2. Okay, so cellular phospholipase A2 is often abbreviated to CPLA2. Right, now what this enzyme is going to do is it's going to migrate to the phospholipid bilayer of the cell. So it's going to go up to the cell membrane. So let's say we have our cell membrane here. Okay, and once it's been activated, and I can't resist telling you that it's activated by binding to calcium. So in type 1 activation, the histamine, or the GQ-coupled ligand, is going to lead to an activation of the GQ pathway, which will lead to the production of IP3, which will cause the release of calcium from the intracellular stores. This calcium is what activates the cellular phospholipase A2. If you want that process talked through slower, then I have an entire video on type 1 activation of endothelial cells, where we go through this at a slower pace. Okay, right, so calcium is going to bind to the cellular phospholipase A2. So here's our calcium ion. It specifically binds to a type of domain known as a C2 domain, but we won't worry about what that means. Okay, so you activate the cellular phospholipase A2 by binding calcium to it, and when you do that, the enzyme migrates from the cytoplasm to the cell membrane, and once it gets to the cell membrane, what it's going to do is it's going to work on a molecule in the cell membrane, and where am I going to write this? Known as phosphatidylcholine. Okay, and I want to explain to you what phosphatidylcholine is. So I'm going to cut this video here, and we'll continue our discussion where we'll look at what phosphatidylcholine is and what phospholipase A2 is going to do to it in the next video.